Hi, I'm Dr. Neela Bakore. In this section, we are going to start with cell. Cell, the unit of life. Let us talk about some historical background, like when the cell was discovered, who discovered it, and all those things. It was discovered by Robert Hooke. He actually observed a cork slide. So when he observed the cork slide, he actually saw hexagonal compartments. Cork, as we know, is a dead tissue. So these walls, they were probably the cell wall, and there was nothing inside because it was a dead tissue. So it looked like a compartment, and that is why he called it cell, which means a small enclosed compartment. Cork slide was observed by him. The cork was of a plant which is quercus. The scientific name of the plant which was observed was quercus. So Robert Hooke discovered the cell. He discovered. But the cell that he discovered was a dead cell. So now the other scientist who discovered the living cell for the first time was Anton von Leeuwenhoek. He observed living cell. So discovery of cell, the credit goes to Robert Hooke because he was the first one who saw the cell, though it was a dead cell. And the credit of observing the living cell for the first time goes to Leeuwenhoek. Robert Hooke is known as, or he has been named as, father of cytology. Cytology is the branch of science which deals with the study of cells. Leeuwenhoek is named as father of bacteriology. Because of the cells which he observed for the first time, he observed bacteria, protists, protozoans, many types of cells. So one important scientist is Robert Hooke, the other one is Anton von Leeuwenhoek. Now let us talk about few more scientists. Another scientist was Dujardin. He observed a fluid-like material in the cell which he called sarcode, which was the fluid-like material. Later on, Purkinje observed the same substance in many other cells and named it protoplasm. Another scientist was Robert Brown. Who discovered the nucleus. So these are important scientists and their contributions in the cell related things. That is, first observed the cell, dead cell. Leeuwenhoek observed the living cell. Dujardin and Purkinje gave the term protoplasm, and Robert Brown discovered the nucleus. Robert Hooke published the entire information about his discovery and observation and everything in his book called Micrographia. So these are some basic historical things which we need to know before we get started with the cells. Now as we are talking of cells, let us talk about the shapes. What is the shape of a cell? There are various shapes which we have observed so far. It can be a cube-like cell, cubical cell, which is seen in germinal epithelium, which we also call cuboidal epithelium. Another shape is long column-like, which we see in columnar epithelium. A slight modification of this is the goblet cell. So this is again a tall columnar cell, the goblet cell. Irregular shape is also very common in cells. The WBCs, that is white blood corpuscles, they are irregular in shape. If we see the smooth muscle fibers, they are spindle shaped. In sponges, there is a special type of cell which has a circular part as well as a small part which appears like a collar and a cilium arising from here. These cells are known as choanocytes. Rectangular cells, the skeletal muscle cells, which can be, if it is a skeletal muscle, it is multinucleated. So this is skeletal muscle cell, rectangular. If we see the shape of RBC, in case of human RBC, because human or mammalian RBCs are enucleated, they are biconcave. That means in the center there is no nucleus, and that's why we see a depression on both the sides. So it is biconcave. But if you see the RBC of frog, then the RBC of frog is bulging in the middle. Reason? There is a big prominent nucleus which is present in the middle region. So this is the RBC of frog. The basic idea is to see how many various shapes are present or seen in cells. These are few examples. There are many more, like neurons, irregular shape. Let us talk about the size. What is the size of a cell? As the shapes are variable, sizes are also variable. The smallest cell is PPLO, or it is also known as mycoplasma. PPLO, the full form is pleuropneumonia-like organism. Pleuropneumonia-like organism. It is placed between viruses and bacteria. This is the smallest cell. The size is approximately 0.1 micrometers. And the largest cell is ostrich egg. We have seen the size of the hen's egg, like normally about 2–2.5 inches. Ostrich egg is about 7–7.5 inches or 9 inches. So it is a big cell. So smallest cell of PPLO and largest cell of ostrich egg. So now the next thing is types. Here we have seen the shapes, the sizes. Now we are talking about the types of cells. We divide all the cells into two categories: prokaryotic cells and eukaryotic cells. Prokaryotic term is derived from two words. Pro means primitive and karyon means nucleus. The cells which have primitive nucleus. Now what is a primitive nucleus? Primitive nucleus is one where there is no membrane surrounding the genetic material. So it is basically naked genetic material which is found in the cell. The example of this is bacteria. So it is primitive. Plus, as there is no nuclear membrane, other membranes are also absent. So, no membrane bound structure, no organelle. Eukaryotic again is derived from two words. Eu means advanced and karyon again means nucleus. So, here the nucleus is advanced. We find these eukaryotic cells in fungi, plants and animals. That means the cells which are found in fungi, plants and animals are eukaryotic cells, and the cells which are in bacteria or bacteria unicellular, so these cells are prokaryotic cells. Now let us talk about one more important historical event, that is cell theory. Cell theory was given by two scientists, Schleiden and Schwann. They put forth three important points which was considered as this cell theory. First point was that all living organisms are made up of cells. It could be cell or cells. So, unicellular as well as multicellular organs were covered under this point. The second postulate or second point which was put forth was that cell is the structural and functional unit. Structural and functional unit of living organisms. Third point was that the functioning of an organism, whatever organism's functions are the contribution of all the cells which make up the body of that organism. So, functions of organism are due to the cells which the body is made up of. Now, this cell theory was modified later on when one more scientist contributed one point here. The scientist was Rudolf Virchow. He added one point. He said all the cells arise from pre-existing cell. So, this became the fourth postulate, and after fourth postulate the cell theory was termed as modern cell theory. So, if we are talking of only cell theory, there were only three initial postulates given by Schleiden and Schwann. Schleiden and Schwann. After the fourth postulate was added by Rudolf Virchow, the cell theory was termed as modern cell theory. The point was the cells arise from pre-existing cells. And so, the cell theory became modern cell theory. So, contribution in modern cell theory is of three scientists, Schleiden and Schwann, which was the original one, and then Rudolf Virchow, who added the fourth point. Rudolf Virchow, he added this point in his own language, and that was written as omnis cellula a cellula. Which was later on translated into English, which meant cells arise from pre-existing cells. Now, in this further part, we would try to see the structure of prokaryotic cell and eukaryotic cells. So, this was just the basic of what cell is and how different shapes, sizes and types of cells exist. So, now let us see the prokaryotic and eukaryotic cells. Let us see the structure of a prokaryotic cell. We are basically talking about the structure of a prokaryotic, or we'll take the example as bacterial cell. So the structure is of a bacterium. There are various shapes in which these bacteria exist. We'll talk about this a little later, but the most common shape is oval, which we also term as rod shaped structure. So this is the outermost membrane, which we call the plasma membrane. Outside this plasma membrane, there is a cell wall. In bacteria, this cell wall is made up of a substance called peptidoglycan. So this outer layer is cell wall made up of peptidoglycan. Peptidoglycan means a part is protein and glycan is for carbohydrates. It's a protein carbohydrate combination. Let us complete this cell wall. At certain places, we find that the plasma membrane is thrown into infolds. There are places where the membrane has foldings, but these foldings are on the inner side. These are known as mesosomes. Mesosomes have respiratory enzymes, so they work or they help in respiration. So basically, they are working like mitochondria of eukaryotic cells. The complete inner compartment is filled with a fluid, which is cytoplasm. In this cytoplasm, the genetic material, this is a prokaryotic cell. So genetic material is without any membrane. That means there is no nuclear membrane. Circular genetic material, DNA. Normally this DNA is found attached to the plasma membrane at one point. It will detach from the plasma membrane when the cell has to divide. So it remains attached to one side of the plasma membrane most of the times. It is circular DNA and there is no membrane. As we said, prokaryotic means pro is primitive and karyon is nucleus. So this is naked DNA which is found. So this is the DNA. This DNA without nucleus is actually known as nucleoid. So in eukaryotes we call it nucleus, in prokaryotes we call it nucleoid, which means basically only naked DNA. Other things, there are no membrane bound structures in here. There are some ribosomes. Ribosomes are non-membranous bodies. These ribosomes which are scattered in the cytoplasm, they are 70S type. When we come to the details of the organelle, we'll talk about 70S and 80S. 80S type of ribosomes are found in eukaryotes and 70S are found in prokaryotes plus the organelle which are there in eukaryotes. In some bacteria or in some prokaryotic cells, there is a circular DNA. This is apart from the main genetic material. This is called plasmid. If plasmid is present, so now here there are two possibilities. Either it is present or absent. So if it is present, then we will call or consider that cell of the positive strain, and if plasmid is absent, then it will be considered as of negative strain. So basically this is just to let us know whether the bacterium is of positive strain or of negative strain. Other than this, there is nothing else inside the cell. These bacterial cells or prokaryotic cells may be flagellate or non-flagellate. If they are flagellate, they can have one, two, many, tuft of flagella, everywhere flagella. Here we are drawing only one flagellum. It arises from the cytoplasm and it's a long thread-like structure. This is the flagellum. So the structure is very simple. During adverse conditions, what we find is outside this cell wall, there can be some jelly-like material, thick jelly-like material, which makes a capsule. This capsule is just a mucilaginous thing which protects the cell from desiccation or drying up. So this would be seen only during unfavorable condition. Otherwise, only the cell wall and plasma membrane. Now when we talk of prokaryotes, or when we come to kingdom Monera, we would talk about gram positive and gram negative type of bacteria. At that time we will also discuss what is the difference in the cell walls or the layers of the cells and why it is a gram positive or a negative. So this is a basic structure. Now let us come to eukaryotic cells, and we will be drawing two together. Eukaryotic cells. As we said, plants and animal cells are eukaryotic. So we will draw one plant cell and one animal cell. So simultaneously we will also be able to understand the difference between the two cells. Plant cells are normally of fixed shape. They are either hexagonal or polygonal or squarish. We have picked hexagonal cell here. This outer membrane which we have drawn is the plasma membrane. In case of an animal cell, it can be of various shapes again, and here we are choosing some oval type of a cell. So this membrane is plasma membrane. Common thing in both the cells, animal cell as well as plant cell. This is a plant cell. So let us write down plant cell here and animal cell here. In plant cell, outside the plasma membrane, there is one more layer which is made up of cellulose, and it is called the cell wall. Cellulose is a polysaccharide which makes up this cell wall, which is found in plant cell. It is absent in animal cell. So this is one distinguishing feature between plant cell and animal cell. In prokaryotes, there is cell wall, but that is made up of peptidoglycan. So this cell wall made up of cellulose, not present in animal cells, only in plant cells. In case of plant cell, the largest structure that we see inside is a big vacuole. This is the biggest structure which is visible in a plant cell. So this large structure is vacuole. In case of animal cells, there are vacuoles, but they are smaller in size. There are many more in number, but smaller in size. So this structure is vacuole here. Because of this large vacuole in a plant cell, the nucleus gets shifted to one side. So in plant cells, we find that the nucleus is shifted to one side, whereas in case of animal cell, this nucleus is normally centrally placed. It is large and is centrally placed. The nuclear membrane here, and here in the plant cell we see the nucleus is slightly eccentric, it is slightly on one side, whereas in case of animal cells it is centrally placed and large. So in plant cell the largest structure is vacuole, whereas in case of animal cell the largest structure which is visible is the nucleus. Inside the nucleus there are chromatin fibers, nucleolus, same is the case in animal cell, chromatin fibers, nucleolus. Normally the number of nucleolus is 1. So this entire structure is the nucleus. Chromatin fibers. Now inside the nucleus there is again a fluid which is known as nucleoplasm. Here also nucleoplasm. So in plant and animal cell the basic structure of nucleus is same because it is a typical eukaryotic cell with a distinct defined nuclear membrane and all other organelle. Now the other organelle which are present in these cells, one is mitochondria. So mitochondria is present in both. In case of animal cells also the number of mitochondria can be variable depending upon the activity level of the cell. If the cell is more active it would have more number of mitochondria, if it is less active it will have less. Plant cells are less active as they don't have to move from their place or the metabolism is little slower, so the number of mitochondria here are less as compared to the animal cells. So this structure which we have drawn here is mitochondria. Another important structure is Golgi complex. In case of plants, those Golgi bodies or Golgi complex is known as dictyosome, and it can be slightly away from the nucleus also. Golgi complex away from the nucleus also, the number can be more than one Golgi complex can be there. In case of animals, normally the number is 1, and whenever we draw Golgi complex we have to remember that the convex side should be facing the nuclear membrane. Why this happens, that we will study when we come to the organelle. So here also we have shown the convex side towards the nucleus and concave side towards the plasma membrane. Another structure which we find only in plant cell is the plastid or chloroplast. Chloroplasts are the structures which have grana, stroma, and in those thylakoids of grana there is this green pigment called chlorophyll. So this is the chloroplast. Exclusively present in plant cell, these are not found in animal cells. So far there are two differences. Plant cells have cell wall, animal cells don't. Plant cells have plastids like chloroplast, animal cells don't. Now let us come to animal cells, a structure which is present only in animal cells. That structure is known as centrosome. Centrosome has two centrioles. This structure is again closer to the nucleus and is found only in animal cells. So right now we have three differences: cell wall present in plant cell, plastid or chloroplast present in plant cells, and centrosome present in animal cells. These are the three differences so far. One more structure which is seen, they are spherical structures filled with enzymes. These structures are known as lysosomes. The number of lysosomes are more in animal cells as they have to digest food, whereas in case of plant cells, the number of lysosomes are less because they synthesize their own food, but they will be required to digest the food when they store it. So, they do have lysosomes, though the number is less. So, now the space which is here is filled with a fluid which is cytoplasm. In case of animal cells also, we have the space completely filled with cytoplasm. So, this jelly-like substance is cytoplasm present in both plant cell and animal cells. Now, this is the basic structure, and as we have drawn both these structures together, it is easier to see the differences between the two. In plant cells, the shape is normally geometric, whereas in case of animal cells, it is slightly ovoid or circular. Cell wall made up of cellulose, which gives a firmness to the cell, plant cell. No such cell wall present in animal cells. A large vacuole in case of plant cell, many small vacuoles in animal cells. Nucleus eccentric, shifted towards one side because of this vacuole, whereas in case of animal cells it is centrally placed. Other organelle like mitochondria common, Golgi body common. Plastids or chloroplasts present in plant cell but not there. One more organelle which we need to draw is endoplasmic reticulum. It is a network of tubules which normally runs between the nuclear membrane and the plasma membrane. There are two types of endoplasmic reticulums. One is rough, which we call RER or rough endoplasmic reticulum, and other is smooth. So, we will draw one of both. So, normally this is drawn in the form of a tube like thing, but it is not exactly a tube. When we come to the structure of the endoplasmic reticulum, we will see the exact structure. And one more which we can draw here. Now, what makes one smooth and other one rough is presence of ribosomes on it. If the membrane is studded with ribosomes, we will call it SER, that is, this will be called RER, rough endoplasmic reticulum, and the one which is without the ribosomes is the smooth endoplasmic reticulum. Same would be in the plant cell. So, here again the endoplasmic reticulum, a network of tubes, and let us draw one more. The one which is studded with ribosome, that means ribosomes are attached on its surface, is the rough one, RER, and the one which is without ribosomes is the smooth ER, smooth endoplasmic reticulum. So, this completes our structure of plant and animal. So, in eukaryotic cells, we have seen the structure of both the types of eukaryotic cells. The fungus or yeast, for example, is pretty much similar to this. Basic structure remains the same. So, now we have three cells to compare. One is prokaryotic and these two eukaryotic cells. You see, in this prokaryotic cell, there is nothing inside, no membrane bound structure. There is one exception, that is of blue-green algae, where there are some thylakoids, which we will talk about in kingdom Monera. And in case of plant cell, basic pattern is same except for couple of organelles which are present or absent in either of the cells. So, two types of cells, prokaryotic and eukaryotic. Now, moving forward, what we are going to do is, we will take individual structures and see the details of it. That means we will talk of plasma membrane, then cell wall, nucleus, vacuole, mitochondria, each structure. These are called organelle. So, now let us take a organelle one by one.